All right, so when we're creating an experiment, there's a couple things we need to do. One, we obviously need to, going back to the scientific method, stating the problem. We got to know what problem we're solving. All right, we're going to have to gather info. Basically, all this stuff is going to be involved in our experiment. All right, all this stuff that we've already gone over. This scientific process. All right, but we're going to talk more specifically about the parts of an experiment. So when we talk about, once again, just to recap, a hypothesis, that is a testable prediction. Must be testable. Must be testable. I'm just telling you that because I every year when we do this, the create your own experiment thing, I'll have students say, hey, this is what we're doing. This is what we want to do. I'm like, oh, that sounds cool. And then they give me their hypothesis, and I say, how's that testable? And every year they're like, well, it's got to be testable. It's got to be reproducible. All right. So just to emphasize, when we do this, do your own experiment. When you form a hypothesis, it must be testable. It must be testable. All right. So when you're designing a controlled experiment, all right, factors in an experiment that can be changed are called variables. All right, variables are basically just a fancy word for things that can affect an experiment. All right, so when I say, what are your variables, it just makes me feel smart. But really, it's just like, hey, what are you testing? What are you trying to do? What are you trying to change? What are you trying to measure? What are you doing? What factors play a part in your experiment? All right, so a variable is just something that can affect an experiment. That can affect an experiment. All right. So controlled experiment works when we're changing one variable at a time. All right. We don't want to change a bunch of things. So if I said like my experiment is I want to see how high a ball bounces if I drop it from a meter. All right. And then I want to see how the bounce height is affected when I raise the ball up to 2 meters. Well, I can't raise the ball up to 2 meters and change the type of ball because the second ball might be more bouncy or less bouncy, and it might have bounced more or less anyway. And so it basically, I won't know if the height was the reason why it changed or if it was because of the type of ball. Basically, you don't know what the reason for the change is. So when we're doing an experiment, make sure only one thing changes at a time. All right, one variable changes at a time. In a control experiment, only one variable is changed at a time. All other variables should be unchanged. All right, what we call that is called a constant. And I'll get into that in a little bit as well. But a constant, once again, something that is staying the same. There should be an experimental group and a control group. All right, these two groups, or it could be more than two groups, but they should be identical except for one factor. Because you need a control group, and I'm going to explain this. I don't expect us to get this right away. I'll explain this in more detail. But a control group is going to serve as a comparison. All right, it's going to be the exact same as the experimental group, but we're just removing one variable. Once again, I'll explain that. If you're like, what the heck? Remember, I'm just taking one thing out of the experiment. Variable, fancy. All right, so a couple things you got to write down that you're going to need to know. What variables are kept constant? All right, once again, variable, just a part of the experiment, a thing that could affect the experiment. So we want to keep things constant. You say, well, what's constant? Remember, constant, if I say, hey, I'm driving at a constant speed or I'm walking at a constant pace, that means my speed is unchanging. All right, it's not changing. So a constant variable means we're not changing that part of the experiment. We're not going to change those things in the experiment. So here's an example. So say we have 50 volunteers. We want to test the effectiveness of a vaccine. One group would be the control group. One group would be the experimental group. All right. So the control group would be handed a pill that looks the exact same as the experimental group. They'd be handing the exact same pill. The variables, the constant, the size, the shape, the color, the texture. The only thing that's going to be missing is one group will have, and a lot of pharmaceutical companies do this, they have something that's called a placebo, a sugar pill, and the other group will have the vaccine or the medicine. They don't want anybody to know which group has which because they fear that might throw off the results. All right, and so they give them both the exact same thing and then they test the results. All right, so the control group in that experiment would be the group that did not receive the vaccine, and the experimental group is the one that did receive the vaccine. Still doesn't make sense? That's okay. I'm going to give you a couple more examples. We got to talk about a few more things before we do that, because I'm going to talk about the thing that we're taking away is called the independent variable. All right, the independent variable, these are other parts of an experiment. This is going to be something you have to label. This should be part of your hypothesis. This should be, when you're forming a hypothesis, these two variables should be very much part of it. This should be in your data table. When you change one thing, how does it affect another thing? The independent variable, again, is something that we deliberately change. Because I would say, hey, how does this affect it? How does this factor change something else? So like the ball height example. So if I drop a ball and I say, well, I want to know how much, if it'll bounce higher, if I raise the ball up more, or if it will bounce less. All right, so what I'm changing is the drop height in that scenario. I'm purposely changing something. All right, I'm making, if you think of it like independent, if you're independent, that means you don't rely on anything else. All right, it's something that we are changing, not depending on anything else. It's something that we as the experimenters are changing. The dependent variable, dependent means it relies on something. If you're dependent, like you're dependent on your parents if you don't drive, you're dependent on somebody to bring you to school. All right, whether it's a school bus, you need somebody else. You depend on somebody else. A dependent variable depends on, it's going to change based on the independent variable. How we change one thing, one part of the experiment will affect the other part. So the thing that this is usually the thing that we're measuring. All right, we're going to measure the dependent variable, like in the example, the drop height, how high it bounces, the height of the ball, how high it bounces. That's the dependent variable. That's the thing I'm trying to solve, the thing I'm trying to measure. How high does it bounce afterward? All right, so the dependent variable is typically what we're trying to solve, what we are trying to figure out based on how it reacted after we changed something. All right, so independent variable is something we change on purpose. We know we're going to change it ahead of time. I'm going to drop the ball from 1 meter, from 1.5 to 2 meters, and I'm going to see how it changes. The dependent variable is what we're trying to solve for. All right, and so here is an example of that. All right, this is probably a little bit better of an example. All right, we're going to use plants. All right, we want to see which plants, say we have 100 plants. We're going to see which combination of fertilizer is the most effective to help them grow the best. All right, so what we're going to do, since we have 100 plants, is that we are going to take, and once again, this is an experiment. We'll talk about the different parts of the experiment. We're going to take 100 plants, divide them into 4 groups. Group 1 will just receive plain water. Group 2 will get weak fertilizer. Group 3 will get medium fertilizer, and Group 4 will get a high concentration of fertilizer. All right, so it's more fertilizer than that. All right, and so we're going to see over time for 1 month which combination is the most effective. Now, the group that is without, if we were to go through and label these, if we go through and label these, what would be our independent variable? Well, what are we changing? We are changing on purpose the amount of fertilizer. So that's our independent variable, because I'm changing the amount of fertilizer we have. The dependent variable is how much the plants will grow. That's what we're going to measure. All right, so and then this group, this right here, this group 1, that's going to be our control group. That's going to be, it's not going to have the independent variable. It's not going to have it. We're not going to add any fertilizer to it at all. So this is the group, whenever you get confused, this is always my most frequently asked question, like, what's the control group? It's the group that does not have the independent variable. All right, and then it says down here, the plants will be watered daily over a period of a month. They will receive the same amount of sunlight. They will receive the same amount of, everything else has to be the same. Same amount of sunlight, same amount of water, same amount of plant, same temperature. All that stuff has to be the same. Once again, why? Because if any of those variables changed, we wouldn't know how effective our experiment was. So we had to keep those all, that's the fancy word we're going to use, constant. We got to keep them all the same. Only thing we want to change is the amount of fertilizer. Everything else needs to be the exact same. So when you're doing an experiment, whatever your dependent variable, independent variable is that you're changing, keep everything else the same so we know which one it is. All right, so after a month, you go through and you measure. Plant group 1 grew an average of 15 centimeters. That's going to be our comparison. So this is what it would grow. This is how it would grow without fertilizer. All right. If we didn't do this, we would have no idea. 35, 28, 10. Well, what if without fertilizer it grew 45 centimeters? We would have no idea. It might actually be hurting the plant. We would have no idea. So by just using the control group without the fertilizer, it grows 15 centimeters. And then the weak concentration grew 35. The medium, it grew 28 centimeters, and the high, it actually did hurt. All right, the plant had too much fertilizer. It actually hurt the growth of the plant by using a high concentration. So this hypothesis was not correct because a weak concentration was the best. All right, so they grow the best in a weak concentration. All right, so that would be an example. So that's an example of having an independent variable, a dependent variable, trying to having a control group, using that to compare. That's what a control group is used for, is to compare information, and then using that to oh shoot, sorry, somebody just came in my door. I forgot what I was talking about. Sorry, somebody just stared at me. Anyway, basically what we want to do from there, after we have our conclusion, is that we would use any of the data we would have, we'd put it, make sure we record it in a data table, and then we're going to draw conclusions. All right, so a couple other common questions in science. Why is it important to have large samples? All right, 25 plants, for example. Well, if you just use 1, 1, and 1, well, I don't know, maybe the one plant had a disease that we didn't know about, and that could throw off the results. But if you got 25 plants of each, one plant is kind of withery, well, that plant might have had something wrong with it. Might have had like some kind of a disease, or a insect might have eaten it or something. So it's important to have large. That's why sometimes I'll say do 3 trials, because something weird might have happened during one of them, and if you just say, well, I did one, that's the answer. Well, it might not be. All right. So some important things when you do an experiment, make sure you have a significant sample size. Usually I'm going to require about 3 trials to do something, just to make sure no mistakes were made, no human error was there. Same thing, once again, just doing it many times. See if we have results. What's the importance of a control group? The importance of a control group? Well, what happens when the experimental factor or the independent variable, what happens when it's omitted? Now we can use that to compare. All right, so that's the big thing. Control, compare. We use the control group to compare. How did it compare to what happened? Other important questions you'll be asked: how is a theory different from a hypothesis? We covered that in the last video. Make sure you understand that a hypothesis is an educated guess. A theory is something that is proven based on the best available evidence, is the best possible explanation based on the evidence provided. Independent variable. Independent variable is the one that we change. The dependent variable is the one that we're measuring. All right, it's the one that we're measuring. And then lastly, why do we keep all variables the same except one? Because if several variables are changed, we wouldn't know which one really was the one that was causing the change. All right, so that's where we're gonna stop. So those are just the basic parts of the experiment. You might want to refer back to this video when we actually go to do our experiment. If you're like, what's a control group again? You might have to go back, and you should be able to move through it. Once you've watched it once, you'll be able to move through it without having to. You'll be able to skip ahead and skip back and all that stuff. So there you go.